In this module, we'll talk about ATP. It's a very important molecule. First of all, let me tell you that not all processes in cells release energy. There are processes that require energy. For example, when we are converting monomers, we are linking monomers and creating polymers, we require energy because you're going from disorder to order. That is a process that requires energy. So cells have to compensate the processes that require energy by having some processes that release energy. ATP is a molecule that can store energy in form of high energy phosphate bonds, phosphate groups attached to other phosphate groups. Phosphate groups are negatively charged, so having bunch of phosphate groups linked together is like compressing a spring. Just as spring can contain potential energy, these phosphate bonds also contain energy. The structure of ATP molecule is on the screen. You can see there are 3 phosphate groups attached to each other to the ribose sugar, and which is attached to adenine. This whole molecule we've already seen in one of our previous modules. ATP hydrolysis releases minus 12 kilocalories per mole. That's a huge amount of energy. This energy can be used to make the processes that require energy feasible. Let's look at an example. So for example, processes that require energy, endothermic reactions, for example, building up protein molecules from amino acids. In order to compensate the energy they require for these processes, ATP can be used. We can break ATP molecule, release the energy, use that energy to complete the processes that require energy. So this is a cycle in the cell. So processes that require energy get the energy from processes that release energy when they occur. We have an example of exothermic reaction, ATP, hydrolysis of ATP, ATP combining with a water molecule and releasing an energy of 7.3 kilocalories per mole. I would like to mention, in my previous slide I said that energy released from ATP hydrolysis is minus 12. That's the physiological value of hydrolysis of ATP hydrolysis. When ATP hydrolysis occurs inside the cell, the amount of energy delta G is minus 12. Here in this example we are going to consider delta G, value of delta G as 7.3 kilocalories per mole. Another process in which glutamate is going to glutamine, this process requires energy. It requires 3.4 kilocalories per mole. So where are we going to get this energy from? So these two reactions are coupled. ATP is hydrolyzed, energy released from ATP hydrolysis minus 7.3 kilocalories per mole is used, part of this energy is used to make a molecule of glutamine. So overall the net result is, if we add 7.3 and 3.4, the net result, the net change in delta G is minus 3.9 kilocalories per mole. So we are subtracting basically 3.4 kilocalories from 7.3 kilocalories. So if these reactions are coupled, remember, one is exothermic, other is endothermic. One is giving energy out, other is taking energy in. We can make these processes feasible. Also, I would like to say that there are processes that although they release energy, they do not occur. For example, if we have 600 polyarginines, we have talked about amino acids. Arginine is a amino acid. If we make a protein of arginines, 50% of arginine, polyarginine will degrade. It will take 7 years for these amino acids to be released from this polymer. Although we are going from order to disorder, the delta G is a minus number. This process is extremely slow. This process, although is releasing energy, does not occur very, very quickly. Speed of the reaction is not dependent upon delta G. Speed of a reaction is a different entity, depends upon a different entity altogether. We'll talk about that in our next module. However, here I would like to mention that if we use an enzyme, for example carboxypeptidase, it can accomplish the same feat, degradation of 50% polyarginine molecules in half a second. So the speed of a reaction depends upon activation energy. We will talk about activation energy in our next module.